Assalamualaikum warahmatullahi wabarakatuh. Good morning or good afternoon, everyone. My name is Cheryl, and I will share about my research with the title Molecular Phylogenetic Analysis of Mangifera based on matK genetic marker using an In Silico approach. Indonesia is known as a country that has abundant natural wealth, so it is also called as the highest mega biodiversity country in the world with the biodiversity of land and sea. This is supported by Indonesia's geographical location, which is between two continents and two oceans, and also a tropical country, thus providing an ideal habitat flora and fauna species. Biodiversity in Indonesia is not only economically valuable, but also ecologically valuable. One of them is mango plant that is rich in fiber as an antioxidant and has high genetic diversity. Mango plants are one of the plants that are conserved in the Bogor and Purwodadi Botanical Garden. The high genetic variation of mango species requires variation information about kinship through genetic approach. Because characteristic with morphological method is considered insufficient and can still be influenced by the environment, genetic markers are needed to support these activities. One of which is matK marker, which has a high substitution rate and can identify up to the species level. This research is continued of the in vitro activity carried by Brin with the chairman of MERS Julisasi Trihadia, which has a raw sequence as a final result. The things that became the main basis of this research first is scarcity of specific studies and limitation of the previous methods. With the purpose is to utilize phylogenetic analysis in inventorying the mango genome or Mangifera, as well as building a database of mango species in the Indonesian Botanical Garden based on genetic marker matK using an in silico approach. Now we move to the methods. First, we have material used in this research. One is DNA sequence data from species Mangifera, and two, reference data from outgroup species. We also have tools used in this research: laptop, internet network, Microsoft Excel, Notepad, software BioEdit, software CrystalX, and CBI Blast, software Mega, PAUP, and Triview. We also have a research flow. First of all, we collecting sample sequence DNA Mangifera with raw sequence data consisting of forward and reverse sequence, result from green and glutawaliji as an outgroup, download from GenBank and CBI. We also have to contate the sequence. The forward and reverse sequence are arranged using BioEdit to create a complete sequence, then stored in FASTA format. Next, we have to verify sequencing with NCBI Blast to compare the matching value with GenBank data. We also have alignment, triple alignment using CrystalX to set the sequence of all species including outgroup. And we have to trimming the sequence using Mega, which aims to equalize the size of each sequence. And we also do phylogenetic tree reconstruction using PAU with maximum parsimony method with a bootstrap replication value and edit in Triview software. Last, we have to analyze data of the value of consistency index and the retention index, and also the value of bootstrap and the value of parsimony characteristic from the phylogenetic tree. Now we move to result and discussion. First of all, we have counting result. Result of counting has an average 1018.92 base pair. Characteristic of the matK structure according to all, the length of the complete matK is up to 1500. Now, result of blast. 100% of 38 Mangifera samples show that all samples were within the scope of the Mangifera genomes. Percent identity have a range of 98, 199, which is a high result and can show that the sample DNA sequence has a high level of similarity with the database reference sequence. The similarity level of a sample can be said to be the same species if the similarity percentage is more than 97. According to Herbert et al., that if the genetic identity of a species has difference of more than 3%, then the species is a different species. Alignment results show the nitrogen-based percentage of Mangifera samples dominated by adenine and thymine bases. AT-based dominance show that the gen matK has an AT-rich character, which is a common feature of the chloroplast genome. The high proportion of informative sites show that the matK marker is able to provide a strong phylogenetic signal of distinguish the kinship relationship between Mangifera species, both at the interspecific and intraspecific level. The research of Herbert states that a good DNA marker for phylogenetic analysis must have a balance between conservative area and variable areas to be able to accurately distinguish species. The consistency index value of 0.982 and orientation index of 0.995 shows a very low homoplastion rate. Now we have result of reconstruction phylogenetic tree that this tree divided into two bifurcation clade and one unresolved clade. Clade 1 consists of 7 species with a bootstrap value that varies by 100%, 71%, 63%. Unresolved clade is the largest group that includes 24 samples of Mangifera matK with the support of a very strong bootstrap value which is 100. Unresolved clades include 11 samples of Mangifera matK that have been identified and 10 samples that have not been identified. Clade 2 consists of 7 species with a moderate bootstrap value of 51%. The bootstrap value in phylogenetic tree is used to show the level of trust and accuracy of branching on clade. More higher the value, more higher the level of kinship that is formed. According to Lostari, the value of bootstrap is categorized into 4 ranges, consisting of high range is more than 85%, moderate is 70 from 85%, weak is 50 until 69%, and very weak is less than 50%. From all the results, now we have conclusion that Mangifera sample is secondary data in the form of raw sequence taken from Bogor and Purwodadi Botanical Garden. The result of the counting show that the Mangifera sample has an average consensus length value is high with the best composition adenine and thymine more dominant than composition of cytosine and guanine. Blast analysis show that all samples are verified as the Mangifera genus with a high identity percentage value. The high percentage of variable informative sites compared to conserved sites and the dominance of parsimony informative sites compared to singleton sites show that matK has a fairly large genetic variation and is able to give a strong phylogenetic signal to distinguish the relationship between species. Phylogenetic analysis show that there are 3 clades consisting of 2 bifurcation clade and polytomic clade, which is we call it unresolved, and CI value of 0.982 and RI of 0.995, which is close to 1, showing a very low homoplastion rate and a high character change consistency. So that the resulting phylogenetic reconstruction obtained are valid, reliable, and representative in explaining the evolutionary relationship and grouping between Mangifera accessions. Based on this result, the matK marker is effective in analyzing the phylogenetic analysis of the Indonesian Botanical Garden sample. So, that's all from my research. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh.